Today, as you all asked, biotechnology, we will discuss how better we can put that input. Of course, we are all teaching,
Good
but in
afternoon.
a much good afternoon, ma'am. Good afternoon, ma'am. In a how much better way we can try to put ourselves for the children. That is what today we will be discussing, teachers. This lesson is little complicated. Why I will say complicated in the sense it's not difficult chapter. Definitely, it's normal chapter only, as usual, average chapter only. But it requires the children to learn more in and around the topic, the relevant concepts and all. That is outside the textbook context. They have to learn. Then only they will be able to understand this particular lesson in a very efficient manner. So it requires a little related field knowledge is definitely a prerequisite for understanding this particular lesson, that we need to always keep in mind whenever we are teaching this. Anyway, now term to syllabus, we have this. Definitely we'll try from 21st onwards, we are going to revise the children. Maybe at the time we can use this also. This method we can try to implement. We'll see, we'll definitely get the benefit of it. So first, the students must understand the definition of biotechnology. What is that traditional, classical bio definition of biotechnology? And now, more importantly, the European Federation of Biotechnology, that concept they have to understand in a detailed manner. Usually, teachers, how I teach this definition is, I will keep it definitely aside. I will give the word by word reading will be definitely there, but I will immediately link it with the next concept. Say, for example, microbe mediated process. We are using the cells directly. So using of organisms or the cells or parts thereof and molecular analogs. So either we can use the organism directly, or we can use the cells of that particular organism, or the parts within the cell, or the molecular analogs like DNA, RNA, protein, anything we can use. Along with that, we can integrate the natural science, so that we will get either some product or we will get some service. Not necessarily all the time product only will be the output. Sometimes even the service will be also the output. So natural science, that we need to integrate with the organisms, their cells or the parts of the cell or the molecular analogs. We need to link one by one, teacher. Usually I am doing that. It is really helping me out a lot. Maybe just I am sharing that my experience with you. You can also think on considering that. Usually I will ask this kind of a rapid fire question to the children. I hope everybody is able to see the slide. There are questions. These questions can be asked to the children. Say, for example, yeast is used for making alcohol. Will it come under biotechnology or not? We can ask questions. First, definitely children will say no. Why? Because having already studied the microbes in human welfare chapter, they will think that is a completely different concept. They won't be usually able to link it with the biotechnology. So we need to again refer. See, we are using the organism. From that organism, we are getting the product. So see, yeast is an organism. We are using that an organism, and to you make one product called as this alcohol, ethanol. So can you say whether it is a biotechnology or not? Now 50% of the student will say yes, it is biotechnology. The others still will have a confusion. So they need a little clarity in explaining to them, so that they will also clearly understand it. Then manufacturing a shoe, will it come under biotechnology? Teachers also can put their answer, no issues. Manufacturing shoe, is it a tissue culture? They will know very clearly, definitely no. But they will think, sir, biotechnology, animal parts also we can use. So will it comes under a biotechnology? Definitely no. We don't use any live organism, or we don't use their parts. So we cannot call this as a biotechnology. So second one is definitely not a biotechnology. Then tissue culture, where a live organism is involved. We are growing the organism in a controlled environment. So this is a biotechnology. Making a crop disease resistant, that is definitely a biotechnology. In vitro fertilization, again, we need to give an example to them. It's a biotechnology. Making a piece of furniture where living organism is used, but dead organism. So it is not biotechnology. RT-PCR, we can even implement the current situation. During the COVID-19 test, we are using RT-PCR. Though they don't know about PCR in general, but they will know what is RT-PCR, most of the students. So we can ask whether RT-PCR will come under biotechnology or not. Definitely, yes, it will come under biotechnology. Same way, an activity of involving an enzyme to join two DNA fragments. Will it come under biotechnology? Definitely, it will also come under biotechnology. So, like when we are asking such a, even we can ask a rapid fire questions, teacher. We can ask many shootout questions. Yes or no. The answer is going to be very simple. They need not to say elaborate answer. Only yes or no. Yeast is used for making ethanol. Is a biotechnology or not? One question. Student will in the beginning, definitely they will not agree with us. They will say it is not biotechnology. We have studied in microbes in human welfare lesson. It is completely a different concept. We need to make them at the time. Again, we have to take them back to the definition. We have to make them line by line. See the organism, see the cell, parts of the cell, molecular analogs like DNA and RNA. Now tell me whether it is a biotechnology or not. By repeatedly asking such a question, just I made 8 questions for you, your reference. Like that, we can ask many such questions to make the children to interlink what are all the techniques are there in biotechnology. So this is the first one. We need to know, we need to make them children understand what exactly the definition of biotechnology and what are all the fields are coming under that biotechnology. There is a separate topic is there in our NCERT reader. What is the biotechnology deals with? So in that context, we can explain by asking many shootout questions, teachers. I am already implementing this. Has anybody has implemented this, teachers, already?
Yes, sir, we are using, sir, actually, but not same question, but we are using some different questions.
Okay, okay, sir. Very nice, very nice. So like that shootout questions we can ask. Thank you, sir. So next challenge the students are having, teachers, they have no idea about biotechnology. What happens? They become a passive learner when we are keep on explaining molecular biology, restriction enzyme, DNA. We will cut the DNA. We will run it in the PCR. When we all say that, children are not able to correlate with how exactly the process happens in a regular laboratory. It's there is no wrong in allotting 140-minute just one period or 40 minutes period we can allot for the children to understand the basic equipments in the laboratory, how exactly these procedures and all going to happen in the laboratory. If we make them understand, definitely the students will feel connected to the lesson. They will feel that, oh, this is how the things are going to happen. These are all the various machineries or equipments used in the laboratory while doing the experiment involve biotechnology. This students will be able to correlate with that. So we need to always give that, and one just eye opener, one small idea about what are all the equipments are being used in a laboratory for the conduction of this various experiments involving biotechnology. So we can say that the ELISA washer and reader, the when they are counting the bacterial colony in a petri dish, how it they will be counting how many colonies are there. That autoclave for sterilization, also for boiling, also how they are using that autoclave, the centrifuge. So the various filtration techniques, laminar airflow, all everything will be done in a aseptic condition. For that they will be using laminar airflow, how that will be looking like. What is a water bath? Even in our laboratory, usually school laboratory, water bath will be there. That also can be shown to the children. This idea definitely we need to share with the children so that children will be able to feel connected to the lesson. They will have very detailed understanding. Of course, they are understanding the content, but very passively they are sitting and listening. They are not able to comprehend or correlate how the process will be happening. Their maximum idea will be only the text will be there in the mind whenever they are studying. They won't relate it with the equipment. So we can use this opportunity to make them understand the equipments. how this, they may think like enzyme will be added under the electron. Even when I go and ask the question to the children, they will say, sir, we will cut the DNA using electron microscope, sir. We'll keep the DNA under electron microscope and we will cut. They need to understand in electron microscope, we cannot do that. So how this process and all happening? We can even carry a small vials. We can carry a small test tube and we can go and tell the children, whatever is going to happen there, it's going to happen in the small test tube only. Enzyme we are going to add, that also we are going to add in the small test tube only. It is not like a very large scale process. It's going to happen everything in a small test tube. That we need to make the children to while learning, they need to correlate with how this process and all happening in a regular laboratory. Otherwise, we cannot show them in the lab, isn't it? This one is an equipment. We cannot use it in our school laboratory. So we need to show the different equipments are used. By saying that the basic biotechnology principle or laboratory technique, definitely they will be able to understand teachers. So this understanding is very important for the children. Next also, next challenging one is this the Boyer and Herbert discovery of this enzyme, restriction enzyme. I will not straight away start the topic at all. I never started that. It really helped me now. Now, first I will explain the children about how a bacterium is being grown on a Petri dish. We all have in our laboratory a Petri dish. We can go and take it and we can show it to the children. This is the plate in which microorganism will be grown. We have to show to the children. We need to make them understand how it is growing. It's a very easy. We can take some jelly. We can take some agar, which is available in the shops. We can make it boil and we can just show it a semi-solid liquid, a jelly-like structure. This will be like a matrix. On that, some nutrient will be added. We can ask question to the children, what are the nutrients will you require for your growth? They will say carbohydrate, nitrogen, protein, all these things they will say. Same thing we can say for growing the bacteria also. They require nitrogen source, they require carbohydrates. All these things will be mixed and will be given. Here we need to link the knowledge of lactose. Then see the magic will happen. They will understand the concept of lactose, lactopron also. They will understand the biotechnology principle also. We can say why the lactose is given to the bacterium. We can ask question to the children. What will happen if lactose is not given, or if any other carbon source is not given? What will happen to the microorganism? We need to ask. They will definitely come up with an idea. Yes, it is a carbon source needed for their growth and metabolism, energy requirement. They will come with an idea that we need to first implement. First one is how a microorganism or bacteria is mainly grown in a Petri dish. We need to explain. Teachers, there is no wrong in spending 40 minutes all exclusively for the related concept, not entering before entering into the core content of the lesson. There is no wrong. Why? Because this understanding will make them understand the concept in a very detailed manner. They will be able to correlate very well. So Petri dish, first one, agar, then nutrients, all these things we have explained to them. Now over. Now keep it aside. We'll ask question to them. When we go to the doctor, doctors give us some, prescribe some antibiotics. Why do he do so? What is the need of taking antibiotics? We can ask question to them. They will say antibiotic will kill the microorganism. All these things they will say. Why this knowledge is important is we need to teach them antibiotic resistant. There is a topic you know very well, antibiotic resistant is there. Most of the children will be facing challenge while we are explaining the selectable marker. Am I right or not, teachers? Most of the children, sometimes they will ask even 2-3 times. Am I right, teachers?
Yes, sir.
Yes. So to overcome that only, now we are going to discuss how better we can put it in their mind. Now, after completing the growth of microorganism, we need to teach them what is antibiotic sensitivity test or antibiotic role of antibiotic. We can directly enter. We can ask what will happen an antibiotic is given to a person. What happens? It kills that particular microorganism. Now you can show the same antibiotic will be placed on some plates where bacteria is grown, and you can see there is a well-like structure where microorganism growth is not seen. What does it indicate? We can ask question to them. This is a particular antibiotic. When we place that antibiotic disc surrounding that to a large sum of area, there is no microorganism growth. What is the reason? We can ask the question to the children. They will give a variety of answer, and we need to bring the answer from their mouth that this antibiotic actually resisting the growth of microorganism not able to grow. When you give the antibiotic, the microorganism is not able to grow. Here antibiotic is resisting the growth of microorganism. Now immediately put the next question. See, here also an antibiotic is there, but surrounding the antibiotic there is no any such a kind of structure. The microorganisms are very well growing there. What is the reason? Here we need to say, here microorganism is resisting the antibiotic activity. The microorganism is resisting the activity of antibiotic. The previous case, the bacteria is sensitive to the antibiotic. In the second case, that microorganism is resistant to the antibiotic. These two terminologies, well before we start the biotechnology lesson, we need to put it into their mind. We need to make them understand how certain antibiotics are resisted by the bacteria, how certain antibiotics are not resisted by the bacteria. We need, if we put this knowledge into the children mind, then while they are learning this biotechnology, whenever the selectable marker and all, they will be just like that will be a piece of cake. They will be very easily, they will be able to understand that, and they will keep in mind without any trouble. So that we can try. This is the basic laboratory technique. As I already have told you, if possible, if it is an offline class, mostly it is not possible to show the pictures, but definitely in our laboratory we have this petri dish and all. We are always we use in all our laboratory schools, every schools it will be there, so we can show it to them. We can take the, no need of any sucrose or any carbon media, only the agar. We can take that agar, we can heat it, we can take it in a liquid form, we can pour it in front of the children, and we can show how it forms a jelly like structure, how it becomes, it solidifies, how it forms a matrix for the growth of bacteria. We can show to them, and we can even show how the bacterial growth is obtained on that, how that bacteria is being inoculated. That word they must know already, inoculum, they know how inoculation is happening. We have to tell to the students by either by the picture, or we can even act like, we cannot grow it definitely in our school's laboratory, we cannot grow it, but we can make them understand how it is actually growing. So these basic things for 40 minutes it will take definitely. If we explain this 40 minutes we spend with the children, definitely it will make the children understand the lesson in a much more broader perspective, and also in a very easily they will understand whatever the forthcoming topics are there, which we proved. Next one is the principle of biotechnology. Here what happens, student will be able to, the definition is very simple only, but we need to ask question, we need to pull the answer from the children's mouth. What will happen if the DNA or RNA is chemically altered? What will happen? Before we reveal the answer of phenotypic changes and all, we need to make them understand what is the meaning of chemically altering the DNA and RNA. Why it has to be altered? By altering, what benefit we are going to receive? What economic benefit the humankind is going to receive? We need to make the children very clearly understand the chemical alteration. We can even pull the words or answer from the mouth of the children themselves. Next one is bioprocess engineering. Here children often fail to understand or don't mention the word that what is the need of maintaining a sterile and ambience environment. What is the need of maintaining the sterile environment? The student must understand. We need to link what will happen if it is contaminated. We need to say the word contaminated. We need to make the children to understand the word contaminated. What will happen if sterile environment is not maintained? What it will lead to? When vaccines and antibiotics are taken by thousands of children and people, after taking antibiotics, something goes seriously wrong with the person, what will happen? What kind of impact it will have? So we need to repeated questions. We need to ask in that so that children will understand what is the importance of maintaining sterile environment. Next one, teachers, usually I won't go in this order of what is there in the reader. I go on my own order, but here just for a discussion, we have made it in the same order. So what are the steps are there in genetically modified organism? Desirable gene. Student will not know what do you mean, what is mean by desirable gene here. They will know it has some benefits. So here definitely we need to recall the previous knowledge which they have studied in Human Genome Project, teacher. If they study the Human Genome Project once, they will be able to understand a little idea about the DNA sequence. And desirable gene, they will have little more idea. Now next one is host. What is the meaning of the host? Which organism can be a better host? All the examples we have to put in front of them, including bacteria, it include plants and animals, everything we have to put in front of the children. And which will act as a better host in biotechnology? What is the reason? We need to ask that to the children. Next one is maintenance of introduced DNA. How it will be maintained? What do you mean by continuous culture means what? That little idea we need to put it into their mind. Next is definitely little this year, last 2 years and this year it is going to be a challenging for us. There is a textbook very important concept that why biotechnology is much better than the traditional hybridization technique. But here, what is the problem is they don't study the traditional hybridization technique. That lesson is not there. The germplasm collection and all, we don't have that this year. Last 3 years, we don't have. This year also, we are not going to have. So this traditional hybridization, students will have a doubt that why traditional hybridization is not so better compared to this. Here, we can do two things. One is Mendel's law of inheritance, we can interlink. We can explain little about Mendel's law of inheritance, how he has obtained that hybrid. That we can explain. One. Second thing, we can even relate why that apomixis is better than high yield variety seed, which we have studied in taught them in the third lesson. Second lesson, sorry. Second lesson we have taught them the apomixis seeds are much better than HYV seeds, that high yielding variety. So that concept also we can link here a little so that they will understand that lesson also very well. This particular lesson also very well, they will be able to understand. Traditional hybridization, they will be more clear in that case. Next, the introduction of this DNA, the second step, how it is introduced, the plasmid concept. Anyway, one more area also it is there. So the plasmid, they have forgotten that plasmid very long back because they have studied only in the 11th standard. They studied in that cell chapter only. They last studied about the plasmids. After that, they don't remember. So we need to again take them back to the 11th standard once again. A quick recalling of what they have studied in 11th standard, we have to put there in mind how the plasmids, what are all the other characters controlled by the plasmids. We need to make them understand a little. We need to definitely recall that so that the children will understand plasmid in a very nice manner. Definitely this and all, it will take time consuming topics only. Biotechnology is time consuming topic only. But when we spend our time productively in these areas, the later part, we don't have to spend too much of time. We can complete in a very quick manner. We can complete the other topics, whatever the advanced topics are there, that we will be able to complete in a very quickly. So next one is origin of replication. It is not a new topic to them. Usually we teach them during the molecular biology itself. We teach them generally. You also will be doing definitely like that only. in the molecular biology itself, while we are teaching about replication topic, then and there itself we have to explain what is that origin of replication. And also we need to say the copy number also. There itself we can say, if we didn't say there, at least here we need to say how it helps in controlling the copy number, we have to say. And after adding all this thing, how the DNA has become a part of a alien other vector, how it has become a part of it, and in a host, how the host has become a transgenic one, that we need to explain a basic idea. Now next one is Stanley Cohen and Herbert Boyer's experiment, a very important 3-mark questions. In many time it is asked in the exam, generally just like that it used to be asked in the exam. What is that Stanley Cohen experiment and Herbert Boyer experiment? We need to explain. Generally, I will never start with that Salmonella typhimurium topic. I will start with E. coli first. I will ask them, E. coli bacteria is a gram-negative bacteria. It's a rod-shaped bacteria. It commonly lives in the intestine. Now, I will ask them, the E. coli do not have any antibiotic resistance. Why it does not have antibiotic resistance? We need to ask them question. They need to understand that antibiotic resistance also genetically controlled. That idea the student should have understand. So first we need to explain the children that E. coli is an antibiotic, doesn't have any resistance to any antibiotic. They don't have such a gene which can resist the antibiotic. First point, this has to be said. Next, we need to say the relative of that E. coli, Salmonella typhimurium, which has resistance to the antibiotic. First, we need to start that E. coli do not have this resistance to antibiotic. Second point, the Salmonella typhimurium, which is more or less related, they have the resistance to antibiotic. Now, from that Salmonella typhimurium, we are going to take the gene which is responsible for antibiotic resistance, and we are going to transfer it into the E. coli, so that using a vector, so that E. coli will also become antibiotic resistant. When we say this to the children, they will very well understand. Otherwise, the students will have a lot of doubts in understanding this particular topic. So first line we have to explain them about both the bacteria, which one has the antibiotic resistance, which one do not have, what is the reason. Second thing, how come that E. coli can also be made a antibiotic resistant bacteria? How can that be achieved? So second point, when we explain that, student will definitely will not have any doubt. Your class will be definitely, they will keep it in the mind. In exam also, they will be able to write it very well, whatever the question is asked. Next, various tools used in the next challenging and very elaborate topic. It is a very long topic. It starts with the restriction enzyme, the vector, then competent host. This covers a lot of topics. That's why what I do, I won't enter into this lesson for this topic first. We'll go with our own order. Usually I do that. Maybe most of you may be following the same thing only. We'll directly enter into isolation of DNA, because that is going to be the first step. So first we'll explain isolation of DNA. Then we'll ask them, okay, now we have isolated the DNA. What are we going to do with this particular DNA? We need to put a question to them. DNA is isolated. Even practically also we have, maybe we might have completed the practical by the time. Then we can even practically show that we have isolated the DNA. What are we going to do with that next? We can say that. So that students also will be along with us, they will also be traveling and they will be explaining. They will understand very well how the DNA, what is the fate of the DNA, what are the process it is downstream it is going to pass through, that the student will be able to comprehend very easily, teachers. You can also try it if at all not tried before. But now for simple discussion sake, we will go with the book order only. We are just going. Then the restriction enzyme. Here restriction enzyme usually student will have in only one area doubt. Doubt also they will not have mostly. We cannot say it's a doubt. It's like clarity, content clarity they may require. That's all. So what is the restriction enzyme? Why it is given the name restriction enzyme? That is very important.
Bacteria can able to produce this kind of enzyme
Very
from
nice, ma'am. Two things it will do. One is a methyl group, it will add to its own
cell group.
DNA, and it will produce the one more enzyme, the restriction enzyme.
Enzyme.
Yes. Why it produces the restriction enzyme? What is the need of that? Can anyone say? Actually, teacher, we can even explain this. We can explain this topic, when we are explaining that Hershey and Chase experiment, there itself we can explain, put an idea to the children. Now you may think why the bacteria produces that restriction enzyme. Anybody can say now. Sir also can join.
Replication is there something.
We can link with the children. Even children, we can ask question. This enzyme is produced to avoid something which you have studied in the Hershey and Chase experiment. That we can say. Yes, Amada ma'am want to say something? No. Actually, it is produced to restrict the growth of the bacteriophage. The phage will be actually infecting the bacteria. It will be injecting its own DNA into the bacterium to use the bacterium as a viral factory. To avoid that, the E. coli produces an enzyme. That enzyme will go and break the DNA of this particular virus into small fragments, so that it will never become a part of the host DNA and it can never multiply. So restriction enzyme, the children must understand the word restriction. Restriction means what? Stopping. When a virus is injecting its DNA into the bacterium is trying to stop the DNA being integrated with its own DNA. So it's restricting the DNA to get integrated. So the student must definitely understand the term, why it is given the name restriction enzyme, because it restricts the growth of or the DNA of the particular virus. So they must definitely understand. Yes, ma'am. So they must understand. So it is given by that scientists when they have discovered like that only. It is restricting the growth of the bacteriophage in the E. coli. Two enzymes were there. One enzyme will add to methyl group to host DNA. Why it will add methyl group means the students will come to question actually. There are at least out of 40 students, one student will ask the question. Ma'am, sir, if the restriction enzyme can break down the DNA of the virus, why can't it break the DNA of its own? Why it won't do that? They will ask definitely the question, isn't it? At least one student also they may ask, what answer shall we give if such a question is asked?
It add methyl group.
Yes, very nice, ma'am. It adds methyl group to its own DNA so that the restriction enzyme will not harm its own DNA. It will only go and cut the viral DNA only. It will go and cut. Student must have the clarity. Thank you, teacher. So yes, so that is the one. Next one, naming very easy. Students will know just like that. They will write all this thing. This only memorizing. They don't have to write that name of the entire bacterium or genus name and all. They don't have to write. Only they have to write what does it represent. Only they have to write. So it is much easier for them. They will definitely do that, I believe. So difference between endonuclease and exonuclease. Here, teachers, we have to put a lot of question to them. Until there is no more question can be asked, we need to ask a lot of questions to them. Say, for example, we can ask, what will happen? Can we use, first of all, the exonuclease on the circular DNA, plasmid vector? Can we use it? How is it possible? what will happen if a exonuclease is used on a normal that is a DNA, that is straight DNA, or a circular DNA? What it will happen? What will happen if you use the endonuclease on a linear DNA, and when you use it on a circular DNA, what are the differences will happen? What are the things you will see? We can ask. And you can say that when a person taken a plasmid vector in a gel electrophoresis, I'm talking later also this question can be asked. The person is adding one restriction enzyme. At last, person is getting only one large band only he is getting. There is no small bands are there. What is the reason? We can ask n number of questions so that children will end up with a saturation point that whatever the question hereafter they will be asking, they will be able to answer very, very well. That clarity we have to give it to the children. Exonuclease, endonuclease, exonuclease where it will cut, endonuclease where it will cut. What do you mean by specific recognition sequence? That is a one key word. Students are writing a story for 1.5 pages also they will write, but they will fail to mention the keyword. When the keyword is not there, we cannot give marks to the children. So we need to make the children to understand how better they can write in a very sharp and crispy way, but still they will get the full marks in that particular answer. Then what was the first endonuclease discovered, isolated? That also they have to know, memorize only. And what is the recognition sequence? This question will come. So this definition, the students must memorize definitely. There is no option. They must definitely memorize what is the recognition sequence. The recognition sequence, they must a 6 base pair sequence, how it is recognized and how it cuts, how the purine and pyrimidine bases are coming opposite to that, how it exactly makes a cut there. The children must memorize this particular definition to the. Yes, ma'am. Amuda, ma'am, or you want to say? Do you want to say something?
No, sir, no. No,
The
nothing.
mic, please. Mic alone. Turn off.
Okay, sir. Okay.
The next one is palindromic sequence. When we say palindromic sequence, we give an example like Malayalam, Madam, and all. Student will understand very well. In exam, when we ask about palindromic sequence example, they will write Madam, Malayalam. That only they will write it. They will come and ask for the mark.
Manorama can be written now, sir. Manorama.
Yes, ma'am. But in exam, that is given only to make them understand what is that, ma'am.
Yes, yes,
But in exam, they should definitely write a sequence name only. They have to write a nucleotide sequence only.
okay,
But
okay,
students are very smart, isn't it? They will write only the examples. They will very well. They will write and they will ask for the marks also. They will forget that. So we need to put them in the clearly, and that's why usually I don't give that example itself. The Madam, Manorama, Malayalam, this and all. No need to give palindromic sequence directly. We can put it either side on the both the strands of DNA. It will read when we keep that position of the strand in the same position. Either side when we read, same nucleotide sequence will be there. That's enough. No need to give the example of all this thing. Only students will keep the example in their mind. And what is the recognition sequence or palindromic sequence of this particular ECO-R1 that they have to memorize? The ECO-R1 by heart. While writing, we need to make sure they must mention the template stand and this both the stand polarity they must mention. 5' to 3', 3' to 5'. Correctly, they have to mention. Repeated conducting test repeatedly, we can put that in practice so that children will never ever forget that. They have to write. If they don't write the polarity, reduce the mark in the test. We tell the children that it is because of so and so reason only it is reduced. So next time you have to change it. So let the children put it in the practice. They have to mention the polarity also of it. Even or 2-3 for safety point of view, they can learn 2-3 enzymes name and their recognition sequence. They can learn. There is no wrong in learning. Next one, very important 2 questions. One is the action of restriction enzyme by taking example of ECO-R1, and second one is the The another one, diagrammatic representation of rDNA technology. These two diagrams are very must for the exam, as we all know that repeatedly in exams also this kind of questions are asked. So we need to make them. Here what will happen, whenever they are writing, two things they should never forget. One is this sequence should be correct. GAATTC, that sequence they should write correctly. And second thing, they should never forget to mention that what are sticky end. What are sticky end? Definitely they must be able to define. Even it can be said like it's a overhanding stretches of the DNA strand. That can make a stick. Why sticky end we call? Can anyone add, teachers? Why do we use the word sticky end? We can say it as a single strand or something like that. Why we use the word sticky end?
That hydrogen bond will be hanging also due to that. When we
Yes,
cut.
ma'am. Yes. Hydrogen bond, how, why? Still why it is called sticky end? The hydrogen, because of hydrogen.
Again, when the one more sequence, when just vector and foreign DNA both they were cut with the same restrictions instead.
Very
So
very
when we
nice.
we can connect. It can be able to connect with the help of that hydrogen bond. It's easy for to connect.
Very nice. We can give a little idea. Sticky note. Sticky note, what happens when you remove the page, it will be always sticky. We can make it immediately paste on any surface. Same way, hydrogen bond is ready. Already it's been broken now. Now hydrogen bond is ready to find its pair. It's eagerly waiting. So whenever another pair is given, it will immediately go and join opposite. That is complementary nucleotides are given. It will immediately go and paste with that without much energy requirement. It will paste and it will form the double stranded DNA. This we need to make the children sticky. Sticky here means already the hydrogen bond is waiting for some its complementary nucleotide to be there. When a complementary nucleotide is given in the presence of a DNA ligase, it will immediately bind with that. So we call it a sticky end. That student has to understand. Of course, they don't have to write it in the exam, but they must understand that content. Next one is the diagrammatic representation. Kindly teachers, make it 10 minutes test also you can conduct, 5 minutes test you can conduct. They must understand how to draw diagrammatically, not using any verb except in the diagram. They must be able to write this diagram very neatly with the proper labeling. Whatever is given in the book as it is, they must be able to write, and they must be given the marks for that. That practice is very much needed for the students teacher. It's a repeated question. Even for now board exam also, we may expect such a kind of questions. Next one is the diagrammatic representation of that rDNA technology. Here what will happen, student will understand the diagrams and all. It's very easy diagram. They will draw it, foreign DNA, circular plasmid DNA. Again, while saying itself, say that linear foreign DNA, circular plasmid. When we use the word again and again, it will record in their mind in a very efficient manner. They will understand very well. But what they do mostly, some points they miss out. Say for example, same restriction enzyme. We need to make sure whether they are using this keyword or not. They must use this keyword. If they are not using this keyword, we need to reduce the mark. We need to make the children understand this is a very important term. It has to be mentioned there. Next one is a DNA ligase. This they have to mention. Next, very importantly, they must mention the transformation. These three keywords, they must never forget to mention. So we need to repeatedly make them write it or understand that these keywords and all must, and they must write the keywords without any doubt. They must write it. Next one is, only can we use only plasmid as a vector? Why can't we use the bacteriophages as a vector? Or how can we use the bacteriophages as a vector?
There we can add blunt end also. When we teach them about sticky end, in case the restrictions in cuts at the center, if it is not cutting away from the palindrome, what kind of ends will it produce?
Yes, ma'am. Blunt end and difference between the blunt end, yes, very nice, ma'am. blunt end and sticky end, we need to give the idea to the children. Usually they won't have that. They don't
Yes.
have the idea because in the textbook only the sticky end is given. Usually in the NEET and all, of course we do teach, but in the textbook we won't teach. But we need to teach to every children what is the difference between blunt end and what is a sticky end. Very nice, ma'am. Thank you. Thank you so much. Very nice. Next one is, why can't we use other things? Okay, is it possible to use? Children will always have a doubt. Bacteriophage is something that phagocytes the bacteria, which actually infect the bacterium and will use it as a factory to multiply. How come we use the bacteriophage as a vector? Students will have the doubt. So we can say that the phage virus only, when it infect that bacterium, now instead of delivering its harmful effect to the bacterium, it is going to deliver our choice DNA, because before that we are going to add our choice DNA into the bacteriophage virus. We need to explain to them so that they will have a clear understanding, because sometimes there is no example in given book, means in a hurry we are all having that pressure to complete the portion on time. Exams are always in the queued up. Next another exams are there, so we are all on the same boat only, but still we cannot compromise on the understanding of the children. So we need to explain them how that a phage virus can be used as a vector. Little a diagram, how long? Hardly or 5 minutes. Whether the children are ready to study or not doesn't matter, it's up to them. We can explain them, at least the toppers, they will make use of it and they will learn in a better way. In exam, any questions are asked, they will be able to answer. Next one is the copy number. Why we should always prefer the plasmid with a high copy number? Here itself we need to explain to them. There itself we can even I relate it with the quantity. One plasmid give 1 gram of protein. If I have 50 plasmid, how much grams of protein will be yield? So which is better? Which shall we go with the 2 copy number, or shall we go with the 50 copy number? Which one will be better? Like that, when we ask question, students will store that in mind. The copy number should be always high. Then a high copy number is equal to high yielding capacity of recombinant protein. So this will make them very clearly recorded in their mind. We can use that very well in the classes. Next one is the very important nightmare for the students. Every other topics they will complete. When it comes to the vector, either they will choose the option to memorize it somewhat, those who are good in memorizing. The others will give it in the giveaway. They will give up their effort to learn it most of the cases. Why that happens? Because only because of two things they come to this conclusion. One is the selectable marker cloning site continuation, and second one is the chromogenic substrate, the giving of the chromosome in the presence of chromogenic substrate, what happens to the beta galactosidase and the what color colony is produced, that they have an idea. Teachers, believe me, after having explaining them what is antibiotic resistance and how the bacterium is grown, if two topics are explained to them, they are able to understand this lesson, this particular topic, just like that they are able to keep in their mind. They are memorizing, no need to memorize. If they don't have to give up, they can understand the lesson to the maximum depth, this particular topic, if they know the knowledge of, if they have the knowledge of what is antibiotic resistance and what is how the bacterium is grown, how the bacteria is grown in the laboratory. These two ideas they must have. Then it's a very easy one. So origin of replication, already we have studied, all the students are very clear about that. In exam, there will be question about ROP proteins also. What is the use of ROP? You know where the answer will be found, teachers, for this? In the textbook, it will not, you will not find it anywhere in the content. Do you know where will you find the answer for ROP? Do you remember? You remember, maybe not able to recall, I think.
Yes, sir.
You will find it below the diagram itself. The small caption would be given. In the caption only, they will mention what is the function of ROP protein. They will not, they wouldn't have mentioned anywhere in the text. As a result, student mostly they won't study. Though we say also it will be just skipped. During exam, they will only depend on the textbook only. They won't depend on any notes also. At the time, what happens? They will forget to read that ROP. Below the diagram, there is a caption given. In that caption itself, they have mentioned about the function of ROP, that they have to study. Nothing. The ROP produce codes for the protein, which proteins are helpful for the replication of plasmid. That's all. That is the thing. That is that is mentioned only in that area. Next one is selectable marker. This definitely for teachers, definitely even having 40 years of experience also, teachers when they go and start the lesson, this topic, because learners are new, teachers are old, we are all experienced, we know the content to the maximum, but learners are always new. When they come to that class, they will look at stare at us like as if we are teaching something completely irrelevant to them. They will have the issue. Here I will teach them what is transformation means. Not only transformation I would teach, I will teach what is transduction, what is conjugation, and transformation. All the three words I would teach to the children. Because transformation means what? Uptaking of the DNA from the surrounding of the bacterium. The bacteria has to take the DNA from its surrounding is called as transformation. Teachers can say what is transduction and conjugation. Very easy only, no? Transduction and
Connecting.
Sir, where there is genetic
Yes, ma'am.
recombination, transformation will take place there.
Very nice.
Yes, sir. Genetic recombination.
Okay, ma'am.
Where we
Good.
can see the transformation.
Good, ma'am. Good. Transformation.
Conjugation. Yes.
Yes, ma'am. Say, ma'am.
Transformation connecting of both the organism with the new conjugants.
Okay, ma'am. Very nice. Then, ma'am, one more is transduction.
Transduction can be done with the help of bacteriophages.
Very nice, ma'am. Very good. Thank you, ma'am. Thank you, Shanmugapriya, ma'am. Now only open the mouth.
Sorry, sir.
It's okay. Okay, no issues. Yes, teachers. Transformation is uptaking of DNA from its surrounding. A bacteria will uptake the DNA from its surrounding is called as transformation. Transduction is a method of transfer of viral DNA into the bacterium, like the phage virus, how it infects. At the time, the DNA from the phage virus will enter into the bacterium. This we call it as a transduction. Conjugation, we all taught in 11th standard. Maybe in here in a hurry we may said like there are two different bacterium. F, you remember F minus and F plus. Yes. Two bacterium will be brought together. A pili protein will be linked in between. Through that pili protein, yes.
In paramecium, we can see, sir.
Yes, ma'am.
Through pili, the genetic material will be transferred.
Yes, ma'am. Yes, yes. F positive, F negative. F negative is the one which do not have the fertility factor. F positive is the one which has the fertility factor. That also controlled by plasmid only. Teachers, believe me, this understanding is very important for the children. They will not understand what is the meaning of uptaking the DNA into the host. What is that? Why it has to be done? They will not have the idea. So we need to give all the three definitions at least. Generally, the DNA from the outside, it can be transferred into the host bacterium by generally three methods. One is conjugation from bacterium to bacterium. That is not, we are not going to do. Second one is transduction, where the virus to bacteria it is transferred. That is also we are not going to see. We are going to see that the DNA from the surrounding medium will be uptaken by this particular host, that bacterium. That is what called as transformation. Here, whenever we are making a recombinant DNA technology, two challenges will be there. First one is our choice DNA. You just put the question to the students themselves. Let them say, choice DNA should become a part of the vector DNA so that it will make a recombinant DNA. This is the first one. Second one is, this recombinant DNA or just the DNA should be transferred into the host. There are two challenges. We cannot see it under the microscope and we cannot do this process. It's all mediated in the cell test tube only, in the vials only it is going to be done. How come we know that whether the choice DNA has become a part of the whole vector, and same time how whether the rDNA has become a part of the host? How come we know that? We need to ask the question to the children. You know, you won't believe, there are hundreds of different answers will come from the children. After hearing that everything, we can reveal the answer. This particular selectable marker is helping us to understanding whether the choice DNA has become inside this host or not. That is along with the vector, or only vector has come inside now without a choice DNA. Two things. Third one is either the vector or choice along with the choice DNA, or only vector alone has not entered the host. Three challenges we need to tell to the children. One is only the vector DNA enter into the host. Only the vector DNA enter into the host without a choice DNA, without a recombinant. Only the vector DNA enter the host without a choice DNA. One thing. Second thing is the vector along with the choice DNA, that is a recombinant DNA, has entered the host. Third thing is neither the vector along with the choice DNA or without that, it is not enter into the host. Three challenges are we will put into the children. Now we'll ask the children. Now we are going to link the topic with the what antibiotic sensitivity test. Now put to the children, if neither vector DNA along with the choice DNA or rDNA, both has not entered the host. Now what will happen if you grow the host in the ampicillin and tetracycline containing medium? First question will ask to the children. We have the host did not receive either the rDNA or just vector alone, has not received. Now you grow that bacteria in a petri dish and you are keeping the disc of ampicillin and tetracycline. What will happen? What will be the result? We'll put to the children. Teachers also can say what will happen. Yes, teachers. What will happen? My question is, vector DNA also did not enter, only vector DNA, or rDNA also has not entered the host DNA, host inside the host. Now you are growing that host on the petri dish containing ampicillin and tetracycline antibiotic. What will be the result? Teachers can say. Teachers, it will be very useful to the children. 3 sentences only they have to study. If they study that, they will understand the antibiotic resistant gene very well. You can try your, we will all together will find the answer. You can say, you try. What will happen if neither the choice DNA along with the vector, that is rDNA, or just vector alone, both did not enter the host? What will happen? What will be the result?
Bacteria cannot be able to survive. No?
Sorry, sir, bacteria will not be able to
Survive.
So what will happen in the result? I mean, yes, bacteria will not be able to, it will be killed. There will not be any bacterial growth around the antibiotic disc. Am I right, sir? Teachers,
Yes,
am I right? Everybody, do you agree with me? Teachers, Amada ma'am,
Yes, sir.
Shanmugapriya ma'am.
Yes, sir.
Now,
Yes,
next
sir.
one is vector alone enter the host, but vector do not have our choice DNA. Vector alone has entered the host, but it doesn't have our choice DNA. Now, what will happen if you grow such a kind of host on the medium containing ampicillin and tetracycline? What will be the result? Anybody can say, Amada ma'am, Shanmuga Priya ma'am, can try. Parthivan sir already said the answer. The question is, vector plasmid alone enters the host without choice DNA. Now, such a host you are growing on a medium containing ampicillin and tetracycline. What will happen? Will the bacteria able to grow or will not be able to grow in the both the medium? Or any one medium also can say. It will be able to grow very well in the medium. Why? Because the vector has both the resistant site, antibiotic ampicillin resistant gene also it has, and tetracycline resistant gene also it has. But it doesn't have a choice DNA. So when we inoculate this particular host on any medium containing ampicillin and tetracycline antibiotic, it will be able to grow very, very well in that plate. Why? Because it has the gene for antibiotic resistance. Now, third condition is, now our recombinant DNA, that DNA, vector DNA along with our choice DNA has entered the host. Now again we are growing such a host in, one is ampicillin containing medium, another one is tetracycline containing medium. Now what will be the result? Now what will be the result? Yes, teachers. We can ask children this question. Usually this is how we say, isn't it? So what will be the result? Now ampicillin, you have added rDNA, choice DNA added. So now we have to say that to the children. It is
survey only in the ampicillin.
Yes, sir. Yes, sir. Any?
They can able to survey only in the ampicillin medium. Only ampicillin medium, sir.
Very good,
Not in
sir.
the tetracycline medium.
Yes, sir. Based on what antibiotic we have used, what restriction enzyme we have used, sir. Say, for example, we have used BAMH1.
Yes, sir.
This restriction enzyme, if we have used, what will happen? Our choice DNA would have become a part of this tetracycline containing antibiotic. So it will lose that resistance ability.
Yes,
It will have only ampicillin resistance. So this bacteria will be growing very well in the ampicillin containing medium, and it cannot grow in the tetracycline containing medium. If on the other hand, we are using PVU1 restriction enzyme and we are adding our choice DNA, then ampicillin resistance will be lost and tetracycline resistance will be there. When we grow the same thing on the two different medium, it will not be able to grow in the ampicillin containing medium, but it will be able to grow in the tetracycline containing medium. Teachers, please, 3 conditions they may have to understand. If they understand it, that's all over. This antibiotic resistance fully they are clear. One is vector alone enters the host without a choice DNA. Second one is vector along with our choice DNA enter into the host. Third condition, neither the vector nor the choice DNA enter the host. Host is without the recombinant DNA, foreign DNA. So 3 conditions are there. So the third place, first one is, what is the use of selectable marker? Means they are helpful for identification of the transformants from the non-transformant. So only the transformants we can take and we can continue our research. So children must understand that these are all like a hit and trial method only. We cannot know whether the DNA has become a part of the host or not. Only when we are growing on the antibiotic plate, we will come to know whether it has become a part or not. So children must understand what are all the difficulties are involved in doing these experiments, and how difficult it is for the scientist to produce the results or to produce the answer, children must understand teachers. So I hope I'm very clear now. So now Amada ma'am will be saying, so only for fun, ma'am, you can say what will happen. Vector DNA alone enter the host, ma'am, without a choice DNA. It is to make sure you are there around. That's why. Ma'am, are you there? Okay, Shanmuga Priya ma'am can say.
Yes, sir. It will grow very well in that antibiotic resistance medium.
Both the medium. Yes, very nice.
Both the
Both the
medium.
medium. Yes.
Yes,
Thank
sir.
you. Namaste, Amada ma'am. First time I'm seeing you.
Namaste, sir.
Okay, ma'am. Next one is the cloning site. So here there is no big deal. Only how many for each restriction enzyme, one cloning site only should be there. We can very easily say what will happen. With the example, we can carry some circle objects on a circle rope or any circle objects we can take. What will happen? Same restriction enzyme have two more sites or two or more restriction sites are there. What will happen? We can show it with an activity. We can cut it in two different places. If it's cutting, it will make the entire DNA into the fragments. Then we cannot do our rDNA technology. We cannot do our experiments. So we can put that into the children very well. Every restriction enzyme preferably must have only one restriction site only. Like that, how many sites are there? Like that, one question we ask to the children. Children will say simply one site only. We can say, teachers, how many restriction enzyme sites are there?
There will be 7 restrictions.
Yes, teachers. Eight will be there. 8 restriction sites are. Yes. Thank you, teacher. Yes. Now, second thing is this particular activity of ampicillin resistant, tetracycline resistant, making them to two different plate, like that 6 plates we have to make. Whether vector alone has gone inside, whether recombinant DNA has gone inside, or nothing has gone inside. To check all these 3 conditions, we need to make 6 different plates. 2 for each we have to make. 6 different plates we have to make. We have to grow them. We have to check it. For a scientist, it's a cumbersome process. It's a very difficult process. Student must understand. Now scientists have came with a very innovative idea with just only one selectable marker. Having only one selectable marker and without much hard work, with only one petri dish enough, with that they can say whether we are success in all the 3 aspects or not, we will be able to say. That is called as this chromogenic substrate, giving of a color colony in the presence of a chromogenic substrate. Teachers, please, children must write the word chromogenic substrate in their answer whenever they are writing. So what is that chromogenic substrate means? The same, we have to pull the idea from their mind. It is already deep settled in their mind. We have to pull it out. That's all. Like, they have studied about LAC-OPRON. LAC-Z, they remember. What is the function of that particular gene LAC-Z? We can ask. Yes, we can take that LAC-Z. We can cut that LAC-Z gene. We can insert into the vector as a selectable marker. So what is the job of LAC-Z? The LAC-Z will be giving a colored colony, beta galactosidase will be produced. That enzyme, if that enzyme mixed with a chromogenic substrate, it will give a blue color. The LAC-Z will be coding for the beta galactosidase enzyme. This particular enzyme, when it mixes with the chromogenic substrate, it will give a blue color. If the enzyme is not there, nowhere blue color can be produced. Very simple idea is over, finished. Now we'll tell the children, instead of antibiotic resistant gene, we can use the gene also as this Z gene, LAC-Z gene also, we can use it as a selectable marker. See, there is one LAC-Z gene is there. Now we are going to add our choice DNA in the place of LAC-Z gene. So laxate gene no more be able to produce beta galactosidase enzyme. When beta galactosidase enzyme is not produced, how come that blue color colony will be produced? It cannot be produced, isn't it? We can tell to the children. I am saying the tone of how we can say to the children, teachers. So now when we grow such a bacteria in a chromogenic substrate, we will only get a white color colony. If there is no choice DNA is there, there is no choice DNA, then we will get the blue color colony. If a choice DNA is there in the vector, we will get the white color colony. If there is no choice DNA is there, we will get blue color colony. Now students will ask question, ma'am, what if the vector itself did not go inside the host? What is the answer? Sir, what is the answer? You can say the answer, teachers. What if the vector itself did not go inside the host? What will be the colony color?
White color.
Yes, it will give the
White color.
white color colony only. There it will create a confusion. We need to tell to the children. So teachers, please tell that. Even you can show the pictures. The online education definitely we can do. For offline also, there is no harm in taking some color photographs and we can show it to the children how the colonies will look like. We can ask the children which colony should we color. We should choose blue color colony, which is very colorful. Should we select that for our down processing, or should we select the white color colony? Which one should choose? We can ask to the children. What, Gayatri ma'am? What do you think?
Yes, sir.
Which one we have to choose, ma'am? Blue color or white color, ma'am?
For what to color the colony, sir?
For next downstream processing, ma'am. Next procedures. For PCR. Yes, teacher. Yes, ma'am. Thank you, Sharmaga Priya, ma'am. Thank you. We have to choose our white color colonies
color.
only. We have to choose. Yes, ma'am. Yes, thank you. So next one is students. This and all they will. I hope it's like a piece of cake only. They do easily understand this topic. If you have any difficulties, you can share with us. We can discuss that also. How can we transfer? So far we are studying only about bacteria. Bacteria is the host, which is a very what is that? Very silent creature, which will not cause any harm to us. We keep on troubling that particular bacteria. What about the animals? What about the plants? We can ask to the children. How can we transfer a choice DNA? Can we depend all the time only on bacteria? How is it possible to make a transgenic plant and transgenic animal? We can ask to the children. They will come up with a lot of idea. Some students, those who are over smart, who already have completed the lesson, they will say that we can use that disarmed pathogen method, something they will say. So we can tell the children very clearly. There are many pathogens are there. They have to understand the word what is pathogen. There are naturally pathogen are there for plant and animals. Very important pathogen in the bacteria for plant is, what is that? We can ask to the children. What is that, ma'am? Teachers also can say. Agrobacterium tumefaciens, we can say. Yes. So this particular bacteria is a pathogen to many plants. Same way retrovirus. We can say retrovirus is a pathogen to many animals. Now we take this particular bacteria and virus, we remove the disease causing area, we remove the gene in that desirable gene there, and we will allow them to infect the plant and animal respectively. Now instead of causing infection, they will be delivering our choice DNA, and plant and animal will start producing the benefits for the humankind. Here another way of exploitation. Teachers, I am using, I don't know, everywhere I use the word exploitation. I never say that word, it's an experiment. I always say the children, it's a kind of exploitation, because the plant we are exploiting for our benefits. Animals we are exploiting for our benefits. So there is no harm in, I believe there is no harm in using the word exploitation. So we are exploiting them, we can say. So next one, how this TI plasmid, very important question for the board exam. Mostly it's a repeated question or asked question. The tumor inducing plasmid, which is naturally present in the bacterium, that causes the tumor. From the tumor, the bacteria will receive the benefits, certain chemicals which is needed for the pathogen's growth. They in exam they have asked once, I believe, what is the need for the bacteria to produce tumor cells in the plant? The tumor is produced in the plant to produce certain chemicals. Those chemicals are very important for the growth of this particular pathogen. So we can have to say that we need to underline that. Next, that Ti plasmid we are converting into our choice one by adding our choice DNA into that, and we are allowing that bacteria to infect the plant. And now instead of causing any trouble to that, it will start producing our benefits to the plant, that we can see. Same way retrovirus teachers, please kindly give the example of the current vaccine. Also we can give example how that Covishield has been made. That also we can give an example while explaining about the retrovirus vector. Next very important concept, competent host. What is meaning of competence? What is why we have to make again and again, that question came in exam. What is the term competent indicates? Why we have to make the host competent? This question repeatedly came. So we need to explain to that definitely the student must understand that DNA is a hydrophilic molecule. We need to make the children understand. And the bacterial cell wall is, it's actually lipid bilayer. We have to explain to that. They are selectively permeable membrane, which will not accept the foreign DNA just like that. So we need to make the bacteria to accept the DNA by treating with the divalent cation like calcium, or by heat shock methods like on ice and then heat. By doing that, we can increase the pore size, or we can use different gene gun methods or micro injection. So gene gun mainly used for the plants, and this micro injection mainly used for the animal cells, that we have to say. Next one is disarmed pathogen method. In disarmed pathogen remote, we remove the disease causing pathogenicity from the bacterium, and we are adding our choice DNA. We have so these are all the very important content so far we completed. So teachers, how do you feel now? Are you since morning you are attending the classes? I know someday will be exhaustive, but it's okay. So these are the things. Now, how they are preparing for the exam, ma'am? Are they prepared well for the exam, upcoming board exam?
Yes, sir. Yes,
Physics
sir.
revision is going on.
Physics revision is going on now.
Yes,
Oh,
sir.
that's what biology. Immediately biology is there. 10 days is given to us. But are they
1 day
prepared
one
in?
chapter, sir. 7 days enough, sir.
Oh, 7 days enough, ma'am. Okay, okay.
Yes, sir.
Okay, okay.
There are few students, they are planned to prepare maths that
Yes,
10
ma'am.
days.
Because
So
for
those
max 7.
maths subject. Yes, sir.
Yes, yes. Only 7 days are there. They will think like only 4 or 5 days enough for the biology. Other days they will, because for max they have only 7 days in between, 6 or 7 days. So
Sir,
they
tell
may.
me some thing, what we can give for the slow learners, sir, actually. For slow learners, what are the things we can make them?
Ma'am, slow learners, will call them in the front, ma'am. I'm going to call only slow learners mainly.
Yeah.
Repeat, we know what are the questions are mostly expected, ma'am. That too only for 35 marks they are going to write. Repeatedly will make them see, for example, biotechnology is there, means restriction enzyme test right in front of me 5 times, write test. We right in front of us, we have to make them sit and we have to make them read and write the diagrammatic representation. If it is a human, I have divided the marks wise only. Human health and disease, microbes in human welfare, 14 marks. Definitely they will be able to get 14 marks if they focus mainly on the immunity. 1, 2 or 3 questions will be there. There's a particular lesson. The first topic.
Plasmodium, life cycle of Plasmodium.
Plasmodium, entire disease together, all the disease together. I gave a very short notes to them. We'll make them read and write in front of me. This 7 days also I'm going to do that only. Right. That 14 marks they will definitely, for sure, they will get money. Then last 2 unit, if we focus mainly about adaptation, this various the Allen's rule and all, if we give, and various conservation, this biological hotspot, endemism and all, if we cover, maximum or 20 they will cross. They will definitely aim to cross.
Quadrant evil.
20.
Quadrant
Yes,
evil.
quadrant evil, that and all. 20 they will definitely cross, teachers. So as I already have told teachers, I will always go with this order only. What you are currently seeing on the board, I don't know, NCERT is going in that order. They are NCERT giving in a different, first they are giving a little highlight of everything, then they are entering into that order. So there is no wrong. If we go in the right from the beginning, we can go with this order, there is no wrong. If we go with this 6 order in a proper direction, there is no wrong in that. It will conserve our time in most areas, it will conserve. Instead of starting with restriction enzyme and all, first we'll start with isolation of genetic material. Directly we can start with that. We need to highlight, we can ask questions. A scientist is actually trying to isolate some DNA from bacteria. He added very correctly everything he did. He has added cellulase correctly. He added protease, like that we give the question to them. We'll ask, still he was not able to isolate the DNA. What is the reason? We'll ask question like that. They will think, the teacher said it's already correct, everything added. Then why he's not able to isolate the DNA? What is the reason? They will be thinking. Can teacher say what is the reason? He did everything rightly. He has taken a bacterial cell. He correctly added all the cellulase correctly. He added protease. Everything he did correctly. Still, he is not able to isolate the DNA. What is the reason, teachers? We can ask such question to children. Can anyone give a try?
Denature because of excess of enzymes, sir.
Yes, ma'am. But I said he has taken a bacteria. He has added cellulase enzyme. And he has added protease, I said.
It's not needed, no, sir.
Yes, that the student must say.
Yes,
Yes, ma'am. That student must understand that is not cellulase is needed for the bacteria. It needs lysozyme. The way, since I said the word everything correctly he did, they come to conclusion. They will come to conclusion that in the step there is no wrong. So we need to make them again and again. We need to put that into their mind. Same way, the DNA can be isolated, like practical also we can go hand in hand along with that. Usually we do like that only, I believe so. So that will be done. Then very important question for board exam. Teachers, kindly this children, make the children to memorize and write this restriction, the purification of DNA. It's a very important one. The DNA fragments are separated by gel electrophoresis. This question will be repeatedly maybe asked. So we need to prepare the children for this particular topic. Here the question student must be able to understand. It's a sieving effect, first thing. Second thing, they should say DNA is negatively charged. It will be forced to move towards anode. Second point they have to write. The fragments will be separated based on their size of the DNA. Third keyword they have to mention. Fourth keyword they have to say the smaller fragments will be very farther and the larger fragment will be very behind. They have to say that. Then the dyeing, fifth keyword they have to write about the role of ethidium bromide and UV radiation. And sixth one, elution, but not only with this teachers. Seventh keyword they have to write this eluted, this particular DNA will be used for the further process of recombinant DNA. They have to conclude this point always with this particular thing. Sometimes CBSE they will ask what are the agros means. They are the natural polymers obtained from the seaweed. Those topic also we may have to include.